morning and welcome back to your favorite news station, Lucky Number 13. Today we have a special story for you. How do poison frogs avoid self-intoxication?
Poison frogs have defensive alkaloids that target the nervous system. Epibatidine is a nicotinic acetylcholine receptor, NACHR, that is used as one of their defensive alkaloids, and it shares a binding site with acetylcholine, which makes it hard to evolve resistance and maintain NACHR function.
In their research, one amino acid replacement decreased epibatidine sensitivity, but at a cost of acetylcholine sensitivity. However, additional amino acid replacements saved the receptor functionality. The overall results showed the evolution of genetic changes that provide chemical defense in frogs.
And now on to our frog expert.
Hey guys! Dendrobatidinae, more commonly known as poison frogs, have independently evolved chemical defenses 4 times. The origins of chemical defense typically co-occur with shifts towards brighter physical coloration, which result in aposematism. Aposematic and non-aposematic poison frogs can then be thought of as theoretical evidence for disruptive selection, where the intermediate phenotype is selected against. There are also multiple origins of aposematism within poison frogs, which suggests that switching between the 2 extreme phenotypes is fairly easy.
Neurotransmitters are usually released and used as communication devices for neurons. They move in the synapse in between the neurons and bind to the next neuron's receptor molecules. However, neurotoxins are chemical agents that can affect the process at any point and can cause problems. When toxic animals evolve resistance to their toxins through amino acid replacements, the location is constrained by protein function. This leads to predictable mechanisms of resistance. Resistance to epibatidine requires decreased toxin sensitivity while preserving sensitivity to acetylcholine that interacts with many of the same amino acids, all without disrupting the normal receptor function.
It was hypothesized that the epibatidine-bearing frogs would have NACHRs that resist epibatidine, yet display normal acetylcholine sensitivity, and that the basis of resistance would involve genetic changes in the ligand binding site. To test this, genes in poison frogs encoding the primary molecular target of epibatidine in the brain were sequenced.
Since 2 lineages of poison frogs were detected using epibatidine, 2 origins of resistance were predicted. The replacements for the 4 sites of the beta-2 subunit were near the epibatidine binding site and had unique amino acid replacement in the poison frog. Each replacement has a single nucleotide charge in the first and second codon position. These suggest non-neutral evolution, especially when 5 additional sites were found unique to the poison frogs, but only 1 was near the epibatidine binding.
And here we are today with Georgia Tech student George P. Purdell, who's going to tell us more about how animals become resistant to toxins.
Frogs have developed immunity to epibatidine through 4 different amino acid replacements. Most attempts to reduce susceptibility to epibatidine will also result in decreased sensitivity to acetylcholine because the binding sites are very similar. The solution for the frogs was for the NACHR binding site to require a very specific receptor so that epibatidine cannot bind to it, while still allowing acetylcholine to bind to the binding site. For evolving this ability, it is hypothesized that the first amino acid replacement decreased sensitivity to both epibatidine and acetylcholine. Later amino acid replacements increased acetylcholine sensitivity while still maintaining low epibatidine sensitivity.
Replacement patterns produced by mutagenesis show that all 3 NACHRs with poison frog amino acid replacements were less sensitive to epibatidine than the wild-type receptor. Overall, the replacements showed a compensation for the decrease in acetylcholine sensitivity sustained by the replacements that provided resistance, as multiple human receptors showed normal acetylcholine responses.
Epibatidine toxin resistance evolved in 3 different clades about 5, 8, and 10 million years ago, most likely as a result of toxic exposure, although there could be some other cause. There are 2 suggested pathways for acquiring toxin resistance. In the first pathway, initial amino acid replacements provide a small resistive benefit, but also decrease the sensitivity to acetylcholine. In the second pathway, mutants are already present in the gene pool, and they aid the emergence of resistance without much cost to acetylcholine or epibatidine sensitivity. Thus, through evolution, resistance to epibatidine can develop in a manner that provides a great selective binding site without interrupting its original receptor function.
We can connect back to what we've previously learned about the process of evolution. Evolution is a change in heritable characteristics of species from natural selection that sometimes enables organisms to better adapt to their environment and survive for reproduction. Furthermore, a change in the amino acid sequence can affect protein function, and that can lead to genetic changes.
That's right. Frogs can develop resistance to avoid self-intoxication and defend against predators through interaction of amino acid replacements. Understanding toxic resistance gives us knowledge about protein function and nervous system evolution, as well as the process of organisms acquiring these defenses.
That's all for today. Thanks so much for tuning in.